So third model of the is the deep learning model, is the training process here. And the last one is the binary and the multi-classification as the result. So let's talk about one by one. Here is the methodology of the data augmentation. Of course, we tried several types of the data augmentation methods, but the GAN was the best one for our data. GAN means the generative adversarial network. So synthetic spectral data were generated using the GAN to augment the limited experimental data set was the very good. So generated spectra are not identical, but very closely approximate to the real data. So before the augmentation, the cluster were the largely overlapped and making it difficult to distinguish between the classes. So after applying the data augmentation, clusters became very well separated and also clearly distinguishable. So you can see also loss training here when generating the data augmentation process. Okay, now we shift into the deep learning. So our deep learning architecture has the 6 layers, 2 convolutional and 1 dropout, and 1 flatten, and 2 fully connected layers. So we trained it with the 624 one-dimensional search spectra, so each containing 1,331 data points, which is about 80% of the total data set used for the cross-validation. So first and the second convolutional layers have the 16 and 32 filters and a kernel size of 8 and the stride of 1. So allowing the model to scan the full spectrum and the capture of the fine Raman peaks. So batch normalization and the TAN activation functions, both of them has helped the model learn the nonlinear spectral patterns while avoiding the gradient loss. These functions applied after every convolutional layer. So after the flattening the fully connected layer with the 16 neurons and dropout rate of 0.4 helps to prevent overfitting. And the final layer use the softmax for the binary or the multi-class classification of the SARS-CoV-2 proteins. So we use it here, the ADAM optimizer, and train it with the batch size 32 of the 100 epochs. So we optimized the hyperparameters, determined it and using the grid search in here. So after that, tuned the hyperparameter is here. Also, we chosen the 2-layer, and accuracy was the best one, and 0.96. And also we choose the 8 kernel. It was the accuracy is also very good, 0.68, 98. Also, we choose the dropout 0.4, and the accuracy was the 0.96 also. So the learned and updated parameters during the training are summarized in here in the bottom right chart table. Okay, this slide shows the performance comparison of the classifiers, which is used here. The random tested a random forest, gradient boosting machine, and support vector machine, and the key nearest neighbor, and the convolutional neural network. So those classifiers datasets augmented using the GAN-based techniques. So here, among the models, these models trained on the limited real-time experimental dataset. And in this case, the random forest achieved the highest performance. And accuracy was the 0.92, specificity is 0.6, and the F1 score is 0.95. So this superior performance can be attributed to the RF model's ability to focus on the high intensity Raman peaks. So in the contrast, deep learning model exhibited relatively lower performance with accuracy 0.8 on the specificity of the 0.2. So, but on the augmented data, deep learning shows the much better result here. So this table summarizes the binary classification result for the all models trained on the both of the real and the augmented data. R means the real data, and A means the augmented data. On the augmented data set, deep learning is best. The perfect scores, it reached to 1. So in other side, we tested the models in several ways. Here, the first test, we mixed 22 Omicron positive and 6 negative samples to test the machine learning model. These samples were not used for the training or the tuning models before. As I mentioned before, we separated all data on two sides, training data and the test data. We use the test data here. It correctly identified the most of them, but 4 positive samples were predicted as negative. Those were the false negatives. In the second test, using the same samples and tested the deep learning model. So deep learning model correctly identified all samples here and showing much better performance. In third test, so we this is a little bit this is the special because we used 35 real clinic samples from the 2 PCR confirmed patients. And these samples is collected for the 5 consequent days. And added 6 negative controls, and then deep learning model made only 1 wrong prediction. The result was very good. So now we talked about the binary classification. Now we shift into second stage. At the first stage, we just detected the viruses, and in this stage, we detected the viruses variants. So in this stage, our multiplex assay works by the encoding each variant with the unique Raman reporters. So Raman reporters is the different. For example, the Omicron peak is 380 inverted centimeters. So beta's peak is the 540 inverted centimeters, and the gamma's is 1,336 inverted centimeters. Also, it has previous peaks in here. So we use the same device as Raman spectrometer with the 789 nanometer laser. Also, the data collection is similar to previous. Here you can see example of spectra. So positive. Sorry. Here positive and the negative. This table shows that the number of the samples. So we collected 54 spectra, and from the 28 was the positive and 26 was the negative. So it's the classified by the variants. Okay, now let's move to the machine learning analysis on the variant detection. So data pre-processing included the baseline correction and normalization and also smoothing. So we tested three models: logistic regression. Oh, sorry about. Okay. Logistic regression and random forest and support vector machine. So the method is very similar to previous the first stage, and the models were implemented using the Python's Scikit learn library with the hyperparameters. Hyperparameters also find out and optimize it via the grid search algorithm. Here are the results. AIZoOM achieved the best performance, and it's reached to 90% accuracy and also a very strong specificity. So random forest reached about the 82% accuracy and with the balanced metrics. And the logistic regression LR lagged behind the 73% accuracy, so limited by the small data set maybe. So this demonstrates that the kernel-based method, such as the SVM, are much more effective for the Raman spectroscopy. If I show the result in the different way, this here to further evaluate the model reliability, we used the rock curves and measure the area under the curve. So in here, the SVM scored 0.89 and RF scored 0.86 and LR scored 0.80. So this result confirmed that the SVM provides the clearest separation between the positive and the negative cases, so making it the most reliable choice for our dataset. So now we are working on here collecting more data sets, and also we are underway to developing the deep learning methods for detecting the variants. So finally, I tell you why this research is important with some examples. So I would like you to imagine the handheld device in the airport and the mobile labs, the self-testing center, or the rural clinics that can detect multiple pathogens within very minutes. For example, the airports testing of the passenger could be finished in the 30 minutes. Waiting time can be reduced considerably. Also, multiple search screening possible for the large group of the passenger. For the mobile laboratories, the mobile testing unit facilities to reduce travel time. Portable system can be used anywhere, and the rapid and cost-effective detections can be everywhere. So everywhere, for example, the self-testing centers, testing kits can be stored like a vending machine in the separate environment. And a portable Raman spectrometer connected to the interface can be kept near to storage area to instant measurement. So it can be implemented in the schools, hospitals, banks, in everywhere. So highlight of the proposed SARS-CoV-2 sensor is the one-pot reaction process and no blocking agents and no pre-treatment of the sample. It also the high signal to noise ratio and the specificity and the reaction time is only 20 to 30 minutes. Also, it's portable, very easy to use, and specific skills and specific devices, specific laboratories not required. Also, it's very cost effective substrate fabrication from the modified just DVD substrate. So it will be a very good step to healthcare sector all around the world. To the conclude, a combination of the AI and the biochip can become super intelligent technology to save human lives from the dangerous and infectious disease by diagnosing in the moment. So this is all about the research work to be pursued the global healthcare. So, however, the small data set used to this research, uh, the deep learning SVM models showed the best performance. So it's the proving that this approach can be used for the real world the diagnostics. In the future, we aim to expanding the dataset for the stronger validation, applying deep learning for the automated feature extraction, and extend detection to other viruses like influenza and the dangerous bacteria such as the Yersinia pestis. Our vision must be the universal and portable diagnostic platform. Okay, so thank you very much for your attention and for your time. I'm glad to answer the questions.
Wonderful. This is really great. Thank you so much. Are there any questions? You can please drop your questions in the chat or indicate by a raise of your hand. Okay, I can see a question. The question is, are we ready to go to lab trials now?
In the currently, it's just finished the project that the DVDs and substrate is fabricated in as a prototype in just for the research work. But the paper is published on the scientific reports. We hopefully that it will be penetrate all over the laboratories soon. It's very new.
Are there any other questions? You can raise a hand or write
Oh,
your question in the chat.
I can see the questions on
Okay,
the chat. Yes.
what are the steps?
Okay. So what are the steps in the Mongolia? So the currently is still the Mongolia is using the traditional methods such as the PCR, the lateral flow. But in the Mongolia, so we are just continuing our research work. As I mentioned at the presentation, we are working on the second stage. We are developing developing the deep learning models here. Also, we are continuing the collaboration between the Tsinghua University to improve and collecting more data here.
Okay, great. Um, there are still more questions.
Okay, so about the clinical trace, I actually, yeah, as your introduction, I'm the engineer and the software guy. Just after the starting the project, I had some knowledge about the medicines and the diagnostic tools. So actually, I can't answer your questions about the rules of the clinic trials, but maybe after the publishing the reports, maybe some of the doctors and the medical scientists provides uh, the clinical trial.
We have yet another question by Hannah.
Okay.
It
Can
says,
series coupled with AI detects emerging? Yes,
can SERS coupled with AI detect emerging
it
SARS-CoV-2
Yeah,
variants
yeah,
before
yeah,
that?
yeah, yeah, yeah. Yes, it is. It just takes 30 minutes. It's not detecting the DNA sequence. It's just using the Raman Raman special spectre. It's like a fingerprint. So it's using the different ways. It's not characterized genomically. So main goal and main purpose is just detecting is the sample positive or the negative at the first stage. So we are successfully detected it, and the second stage goal is the detecting variants. So variants is also detecting just in the 30 minutes.
Yeah, I understand that it's not genomically characterization, but just my question is, can we find new variants that we have not yet sequenced it? This is
Yeah,
what
yeah,
I
yeah, yeah.
what I mean. Yeah. Okay.
So here is for example here in
Maybe I
here.
missed it. I'm sorry.
It's
Uh,
okay, no problem. So in here, because I didn't talk about it in this picture. So
uh-huh.
uh, the variants of the COVID and also the flu, influenza A and B, it has the same symptoms. Oh, sorry,
Uh-huh. Okay.
same symptoms. It's very important to detect the variants, which oh, what's happened? Which variants is it? So in here in Raman spectroscopy, there is some peaks. For example, there is no need to characterize the genomically
Okay.
characterized.
That's
It just
nice.
shows the peaks. For example, these peaks means that influenza A, these peaks means the influenza B, like this one, the Omicron,
Okay.
this one is the beta, this one is the gamma. That's it.
Uh-huh. So what is the limitation of this technique? It seems miracle. It
So
seems a miracle.
I can't answer as a limitation of the medical things, but in technically it's opens a lot of possibilities because it's very portable. So we don't need it can be just a small portable device
Okay.
can be detect the viruses with its variants in the anywhere. And
Okay.
technically it's no limitation.
Yeah. That's great. Thank you for
Yeah.
your presentation, Battamir. Thank you.
Yeah, thank you very much. Thank you for your time.
Okay. Are there any other questions? Any
Okay.
questions or comments? Okay.
Guess it's done.
I think you can stop sharing now.
Okay.
Great. So I'd like to thank you so much, Mr. Battamir, for this great presentation. Indeed, this is a great diagnostic tool that is very promising. And like someone mentioned, I think this needs to be rolled out as quickly as possible. It's something that is affordable and it can be used at the point of care. So I think this is something that is I mean this will really be a very important addition to the diagnostic tools out there. So thank you so much. We really appreciate this presentation. And thanks to the Mongolian team, our team in Mongolia, wonderful team doing great work out there. Professor Seren, we appreciate you. Thank you so much. And to the
Thank you,
audience,
Professor.
I'm sure this has been a very exciting lecture. So thank you for joining today. And we wish you the best of the rest of the day or night, wherever you are. Thank you so much, everyone. Bye-bye.
Bye-bye. Thank you very much.